Welcome to EAACI TV. This is a world premiere. First time that at the Congress we have this TV program, and I'm very proud to introduce to you Professor Hugh Sampson from the United States of America, from Mount Sinai Hospital in New York City. Hugh, would you be so kind to introduce yourself a little bit to our spectators?
Okay, so I'm Hugh Sampson. I'm a professor of pediatrics at the Mount Sinai School of Medicine in the Division of Allergy Immunology.
And you are the world champion in food allergy, I dare to say so. And so you gave a big lecture. What are the current most important problems in the field of food allergy?
Okay, so I think you and I have both dealt with food allergy for a while, and I think it's a very exciting time in food allergy because we're now getting to the point where there are some immunotherapeutic methods that may become available in the next year or two, so we can finally start treating our patients instead of just telling them to avoid foods. Also, from a diagnostic standpoint, I think we're getting much better diagnostics that are actually enabling us to predict the outcome of patients' natural history, but also perhaps what's going to happen when they go through immunotherapy.
So you are alluding to not only skin tests, but also in vitro diagnostics, measuring antibodies.
Yes, so we've been involved for a number of years looking at epitope-specific binding to these various proteins, and what we're finding is that by looking at the profile of how a patient's IgE binds to particular proteins, we can actually predict the natural history of that patient, whether they're going to outgrow it or not outgrow it. We're accumulating data. We think we may be able to tell whether they're likely to have a severe reaction as opposed to a much milder reaction, and then also, will immunotherapy be beneficial for them?
So you are doing molecular allergy diagnostics also at the epitope level, at the single amino acids of this molecule.
Yeah, so we're looking at short chains of amino acids and looking at things like IgE, IgG4, IgA, and by using our statisticians that are very facile in big data, they can put together profiles for us that help us be much more predictive.
So this is the diagnostic part, but you alluded yourself to the therapy. Will there be a kind of immunotherapy also for food allergy as we had it for pollen and mites?
Well, we certainly hope so. There are two companies have been developing forms of immunotherapy, one in epicutaneous immunotherapy, which has completed a phase 3 trial and is in the process of filing. Another company has done same thing with oral immunotherapy, and they're in the process of analyzing their data and then going forward for submission. So hopefully within the next 2 years, certainly we will have some form of immunotherapy. But many new things in the pipeline as well, some very innovative forms where we're looking at DNA vaccines, even looking at modified proteins. So I think there's a number of very promising approaches that are going to be coming in the future, but they'll be farther behind.
Because maybe 10, 20 years ago, one could say the field of food allergy is at a level like hay fever was in the 1950s. But this is going to change.
Yes, I actually believe that in food allergy we may become even more definitive, and we'll be leading the way, because I think one of the things that we have is when you feed a person a food, they're either going to react or they're not going to react. That's not always possible to predict how the pollen's going to fly that year. So we have a very specific way of making the diagnosis, and there are a lot of good studies now looking at immunologic mechanisms as well, and much being presented here. So we may actually take the lead.
So you are very optimistic. That's wonderful. I thank you very much for this exciting interview and for sharing your experience with us.
Thank you.